I'm Medwedeff. In this presentation, I will be describing what dynamic graph grammars are and one particular application for cortical microtubules in plant cells, along with the prospects for parallelization and high-performance computing. For the purposes of this presentation, our graphs are spatially embedded in 2D space. I'd also like to take a moment to describe what plant cell microtubules are and what the cortical microtubule array is. In short, microtubules can be thought of as microscopic tubes that give a cell its shape and play various roles in transport. For the purposes of this short presentation, you can think of them as the scaffolding inside the cell. The cortical microtubule array is the collection of microtubules associated with the plasma membrane, and that is basically the part of the cell that separates the interior from the outside environments. The cortical microtubule array plays an important role in cell shape and cell division. So now then, what are dynamic graphs, and more importantly, dynamic graph grammars? A dynamic graph is a graph that can change or be rewritten over time. For example, the animation on this slide shows a graph that has nodes and edges being removed. And as it cycles back through, we see the nodes and edges are added back in. This is what we mean by dynamic graphs and rewrites. Now, a collection of these rewrite rules is what forms a graph grammar. It's a declarative set of rules that tells us how the graph can be rewritten. These grammar rules can be either deterministic or stochastic. We can then use the domain-specific knowledge. In our case, that's the dynamics of cortical microtubule arrays in plant cells to define a model. The model is the set of rules we defined and is mapped into a master equation. A master equation is a set of first-order linear differential equations governing the time evolution of joint probability distributions of state variables of our dynamic system. And the master equation itself is a computationally intractable equation to solve for large systems directly, so we use a sampling algorithm to simulate it instead. So our simulation algorithm is actually mapped into our simulator, and we have so far written a serial dynamic graph grammar simulator in C++17 with minimal dependencies, which makes it portable and accessible. We've even developed our own mini dynamic graph library, yet another graph library, also called YAGL, and this allows us to create and rewrite dynamic graphs in our algorithm. We've even also developed an expanded cell complex, which is a specialized graph for describing the system's geometry and topology. We use the expanded cell complex to help leverage spatial information to accelerate our algorithm. And you'll see on the next slide, we actually get accelerations in serial, but the acceleration that we get in serial can actually even be parallelized. And that's where we find our prospect for parallelization to high performance computing. And we've also heuristically solved subgraph recognition problems for labeled graphs. And we had to solve this because simulating dynamic graph grammars involves these graph rewrite rules. And when you want to rewrite a graph, you have to search for the pattern that you're going to rewrite. And so we developed an algorithm to find these patterns in the larger system graph and use those as the matches that we apply our rewrites to. And we've also developed, as I mentioned, an approximate simulation algorithm, which speeds up the original and exact simulation algorithm. So here is a performance chart of the exact versus the approximate. So as I also mentioned, we use the expanded cell complex, and that is what helps us accelerate our algorithm. It defines that geometry and topology relationship that I mentioned, and that's encoded in these 8x8, 4x4, 2x2, 2x1, and 1x1. Essentially, these are subdivisions of the simulation space. And the original algorithm doesn't actually have any of this, use any of this spatial information to accelerate itself. So on the top line, you see the 1x1 simulation, and it starts getting pretty slow after 8 iterations. The reason why it slows down after a number of iterations is because more stuff in the system is happening. Now on the bottom is the best version of our algorithm running in serial. On the 8x8 subdivision, you can see it's dramatically faster. And it just so happens we can actually parallelize over each of these individual subdomains, so these 64, and that's what will give us future acceleration and parallelization. And finally, this is a visualization of what cortical microtubule array might look like. And the rules that we used in this model are defined by some parameters that we've gotten from different papers and talking to our collaborators. And this is essentially what we see when we simulate this. And the expected behavior occurs for this model over time. A network appears. Thank you very much.